All right, welcome back. This is biology lesson 7.07 on electrophoresis. And what we're talking about today, electrophoresis is the process of separating fragments of DNA using an electric current. So let's write that down. The definition is separating fragments of DNA using electric current. Just focused in a little bit. It's about the same. Well, why fragments? Why can't we just run a whole piece of DNA? And if you remember, DNA has this, the long shapes. We've got the double helix here. Why not just stick this in and separate the whole kit and boodle? Well, it turns out the one piece of DNA, it all run the same length. So when we talk about the reason why these separate, you'll see why we have to break them into pieces. But you'll notice up here at the top, it says mixture of DNA molecules of different sizes. Why are they different sizes? Because we've broken them ahead of time. And so what I'm going to do is I'm going to flip to the next page. I'd like you to go there with me. And we're going to look at restriction enzymes. So tape down my paper. I think the title on this paper is restriction enzymes. Get on screen here. Okay, so we have said before in the past, we define an enzyme as a protein. So we know that enzymes are proteins. And these are proteins that have a specific job. A restriction enzyme will cut DNA. It'll cut DNA only at specific points. So restriction enzymes will cut DNA only at specific points. So if I've got my two strands of DNA, if I unwind them, and I've got strand here and a strand here, and I've got I'm just gonna, we have all of our nucleotides, right? Because remember, DNA is made of nucleotides. I've got G, G, C, C, A, T, and then my strand goes on. And over here, I've got T, A, T, C, C, G, G, TA. So what I'm doing here is I'm keeping in line the base pairing. So if one strand has an adenine, the other strand has a thymine. If this strand has a thymine, this one has an adenine. Well, as you go through, DNA has ends. So this is the 5' end and the 3' end, and this is the 3' end and the 5' end, and they run opposite to each other. And when DNA gets read, it kind of gets read from the 5' to the 3'. So it gets read this way and it gets read this way. And if we look at this section right here, so what we did was we came down and we kind of blew this up. You can see that going 5' to 3', I have GGCC, and over here I have GGCC. So it's a little bit like a palindrome. A palindrome is a word that says the same thing forward and backwards, like Bob, or oh gosh, now I can't think of another palindrome. Think of one in your head. If I read it this way, I read GGCC. If I read it this way, I read GGCC. The restriction enzyme for this will come in and it will cut here. So there is a specific restriction enzyme that cuts the sequence. And there's more than one restriction enzyme. Actually, we've got lots and lots of them, but each one has its own specific pattern it'll cut. So everywhere in the DNA that there's a GGCC, CCGG, this restriction enzyme will cut, and that's how we get our fragments. So let's go back to our other sheet. Pull this one up and get rid of it. So now we put our fragments. We put our fragments into So an electrophoresis unit has a gel. It's got like this, it feels a little bit like finger jello, if you've ever eaten finger jello. So we've got this gel, and inside this gel, we have these wells. So this equals a well. And at one end, we have a cathode, which has a negative charge, and at this end, we have an anode, which has a positive charge. So we put our samples into these wells, and we turn on the juice. These fragments have charges. The fragments have charges on them. Because there are spots on the DNA fragments that have positive and negative charges. And they also have sizes. So using the size and using the charge, these begin to separate. And so what you saw here was the mixture, and now you're beginning to get separation. And it doesn't look too different yet. But as we apply our current and the fragments get to the other side of the gel, what we see is that it takes less time for the shorter molecules to move, and the longer molecules, it takes more time. So at the end, the longer fragments are still back towards the beginning, and the shorter fragments have kind of zipped through this gel. And it creates a separation pattern. So what we have here is a separation pattern. Separation pattern. Usually what we'll do is we'll make this first column what's called a known or a standard. So the first column is a known or a standard. A protein that we know what amino acids it has, and we can measure, we could say, okay, DNA. Fragment X has this length, or goes to this point, and fragment Y goes to this point, and fragment Z goes to that point. And we can use that to compare them. We put unknowns here. So what you're going to do for the lab is we're going to have, you're going to cut, you're going to be the restriction enzyme, you're going to cut your DNA into fragments, and then you're going to run that on an imaginary gel. Your longer fragments will be back here, and your shorter will be here. So you'll have a column for your robbers. We have someone who committed a crime. And then you'll have a bunch of unknown, you'll have all your suspects. And you'll have to use the suspect DNA to figure out who the criminal was. So that's what we're doing in lab, and we're going to do that early next week. But, so our takeaways here. Electrophoresis, it's a way of looking at DNA. So the first thing we have to do is break it up, and we do that using restriction enzymes, and they have a repeating pattern that we recognize. We take that sample and we put it into a well, then we apply current, and the current begins to separate it. The longer fragments hang back, the shorter fragments zip through, and it creates separation patterns that we can use to analyze to see what pieces of DNA we have. And we can learn a lot about this whole piece by looking at the parts. And we'll do practice with that. We'll do the lab, and then we'll do some more practice with that. All right, guys, that's all for today. Thanks for watching.